Hello everyone, and welcome to this audio podcast. This audio podcast as part of the Prompt series. This audio podcast is part of a Ball and Rumble. This audio podcast is part of an Atomic Headline. This audio podcast is part of a Storm Robotics production. In this audio podcast, looking at branched DNA or bDNA technology, and in around this, it is where in this methodology. In this methodology, it is the signal rather than the target, which is amplified, which is amplified. The target nucleic acid is captured to a solid surface through multiple contiguous capture probes. Extender probes hybridize with adjacent target sequences and contain additional sequences homologous to the branched amplification multimer. Enzyme labeled oligonucleotides excuse me, enzyme labeled oligonucleotides bind to the bead DNA through homologous base pairing and the enzyme probe complex, which is measured or determined by the detection of chemiluminescence, and all hybridization reactions occur simultaneously. Through this, then there is also a little bit in around or in around in relating to relating to bDNA or branched DNA. There is then also some bases in around some of the main features of this particular sort of methodology, but also in around a rapid PCR and in-tube detection of amplified products. So some of the main features of the light cycler, for example, generally is that there can be there is the speed in the amplification of the in the in around the detection, around the analysis of the microorganisms, the specificity, the sensitivity, and accurate quantification, accurate quantification, and then in around fluorogenic reporters, there is also known as dsDNA binding agents and sequence specific probes, and then with other PCR related or real time systems that are available, there are instruments such as Techman instruments and iCycler and Smart Cycler, etc. However, there are sort of a varying number of PCR real-time systems that are available. At the same time, in around this is that the the use of molecular amplification methodologies in diagnostic microbiology also is where firstly there's identification and quantification of particularly around HIV, HBV, and HCV sequences. There's identification of high risk infections with molecular and papillomavirus types. There's identification of C. trachomatis and M. tuberculosis infections. There can be detection of microbe specific nucleic acids in dilute solutions such as CSF or cerebrospinal fluid and ocular fluid, and there can be detection and analysis of mutations associated with drug resistance. At present, generally speaking, PCR and NASBA and bDNA are broadly used for viral diagnosis, and LCR, TMA, and SDA have been utilized for analysis of different microorganisms aligned to bacteria. The range of techniques or investigations used and the number of targets examined or investigated in this way is increasing rapidly, or have been increasing rapidly. New new you know new new laboratory investigations include have included real-time NASBA for amplification and detection of RNA targets. So that draws this audio podcast to a close. This audio podcast, as part of the prompt series, this audio podcast as part of the Brawl and Rumble, this audio podcast as part of the Atomic Headline, and this audio podcast as part of a Storm Logistics production.